Welcome to the Deep Dive. We take complex research, pull it apart, and give you the core insights. Basically, we do the reading so you get the essential knowledge. Today, we're diving into a really interesting paper, quite technical, but it could genuinely change how we monitor life in rivers and streams. It's called eDITH, an R package to spatially project eDNA-based biodiversity across river networks with minimal prior information. The lead author is Carraro, working with Florian Altermatt.
And this isn't just, another method paper. It feels like a real shift in using environmental DNA or eDNA, especially in water.
Absolutely. We all know eDNA has been a game changer for finding out what lives where it's quicker, often cheaper, but rivers are special.
Because the water flows, right? So the eDNA doesn't just stay put, it moves.
Exactly. That passive transport means a single water sample isn't just telling you what's right there. It's potentially carrying DNA from, well, the whole upstream area that drains to that point. It's kind of like how environmental chemists can sometimes trace pollution back upstream from just one sample point. It's this aggregated signal.
Precisely. But that potential, it hasn't really been fully unlocked. Most studies would just look at an eDNA sample and say, okay, this species was present here. They weren't really using that upstream aggregated information. And trying to actually model that transport and integrate it properly, that needed some serious specialist skills in hydrology and ecology, not something most field ecologists could just jump into.
Okay, so that sets the stage perfectly for eDITH, because it sounds like it's built specifically to bridge that gap, to get rid of that need for super specialized knowledge. eDITH, eDNA Integrating Transport and Hydrology. Let's break it down. What exactly is this model doing that standard analysis just couldn't?
Well, it directly models the two big things happening in the river. How the water carries the DNA downstream, that's the hydrological transport, and critically, how that DNA breaks down or decays over time as it travels. By modeling both of those, it lets you upscale the data you get from your sample points. So instead of just dots on a map, you can generate these space-filling biodiversity projections, basically a continuous map of probable species presence across the entire river network.
Ah, okay, so it's not just saying species X was detected here. It's trying to figure out, based on the concentration and where you sampled, where upstream that DNA likely originated from. It's like reverse engineering the river's message.
That's a really good way to think about it, yeah. It estimates the shedding rate, essentially, for every little bit of the river network upstream. And it does this using three parts that work together. First, there's a species distribution model, and SDM.
Okay, an STM. What's that telling us in this context? Where the species might be living?
Sort of, yeah. It links the potential for a species to be there and shedding DNA to environmental factors, things like, temperature, elevation, maybe flow conditions in that specific reach. It predicts the likely rate of DNA input based on the environment. Then the second part, and this is really the core hydrologic piece, is a mass balance equation. It basically says the amount of DNA you measure at your sample spot is the sum of all the DNA shed upstream minus what's decayed on the way down.
Right, and that minus decay part is crucial. The further away the source, the less signal you'd expect to see, correct?
Exactly. Distance and travel time matter because the DNA breaks down.
Yeah.
And that leads to the third component, an observational model. This part deals with the fact that eDNA measurements aren't perfect. There's noise, uncertainty. It helps match the model's predictions to the actual, sometimes messy, field data.
And the really practical breakthrough here is bundling all of this quite complex ecohydrology into an R package. You mentioned R, that's the statistical programming language lots of ecologists use. So this makes it accessible, right? A conservation biologist who maybe isn't a hydrological modeling expert can actually use this sophisticated tool now.
That's the goal, absolutely. It streamlines the whole workflow, things like figuring out the river network structure, the basic hydrology. It leverages other tools like the RivNet package to handle some of that automatically. So the user doesn't need deep prior knowledge of how to model the water flow itself. They just need the right input data. It democratizes the process, really.
Okay, let's dig into those inputs and assumptions. What's the fundamental assumption about how DNA behaves once it hits the water in a flowing river, elotic environment?
The key assumption is that these DNA particles are essentially passive tracers. They get carried downstream by the flow that's advection. So what you measure at point B reflects the integrated signal from the whole catchment area upstream of B.
And the model connects the amount of DNA shed to the species itself. How does it make that link?
It assumes the rate of DNA production or shedding is, broadly speaking, proportional to the taxon's density or biomass in that location. More fish, more DNA, basically.
Okay, and the decay, how fast does it assume the DNA breaks down? Is that a fixed number?
No, and this is quite neat. It assumes the decay follows what's called first-order kinetics, a constant rate of decay. But the actual value of that rate, often symbolized by tau, isn't fixed. It's estimated by the model as a free parameter based on the patterns in the eDNA data you feed in.
So the model learns the decay rate from the data itself. That feels more robust than just guessing a value.
It does make it more adaptive to the specific river system and conditions, yeah.
But there's another assumption mentioned in the paper about the hydrology being constant during sampling. How big a constraint is that? Rivers aren't always stable, right? Flows change.
That's definitely a constraint to be aware of. It means, ideally, you want to conduct your sampling campaign during periods of relatively stable flow like base flow conditions, not right after a big storm. If the discharge and velocity are changing wildly while you're collecting samples across the network, it messes up the model's ability to accurately trace the signal back upstream. Consistent conditions are important for that spatial inference.
Makes sense. So stable flow conditions are best. What about the sampling design itself? How do you need to spread your samples out across the river network for this to work?
Well, first, you need decent coverage of the whole network you're interested in. The paper suggests a rule of thumb, maybe aiming for something like one sampling site for every 10 to 20 square kilometers of area draining to it. But the really critical thing is the distance between samples along the river channel.
Why is that so important?
Because of that decay we talked about. If your samples are too far apart, the DNA signal from the upstream site might have almost completely decayed before it reaches the downstream site. The paper gives a pretty stark example. Under typical conditions, like water moving at 1 meter per second and a decay half-life, maybe around 4 hours, if your sites are more than 50 kilometers apart along the stream, you might only have about 3% of the original signal left.
Wow. Okay, only 3% at 50 kilometers. That really hammers home how fast the signal can fade and why you need relatively dense sampling along the river's path, too far apart, and the sites become effectively disconnected from an eDNA perspective.
Exactly. They need to be nested within that decay distance. But on the plus side, the actual data you need to run the package is pretty minimal. You need your eDNA measurements, obviously either concentrations from something like qPCR or read counts if you're doing metabar coding, and then just some basic hydromorphological data.
Like what? Do you need detailed river maps?
Not necessarily super detailed. You need the coordinates for the watershed outlet, basically defining the area, and then just a single measurement of stream width and discharge somewhere representative. The package, using RivNet, can then extrapolate the likely hydraulic conditions across the whole network from that minimal info.
Okay, so you feed in your eDNA data, your basic watershed info. The model runs, accounting for flow and decay. What's the main thing you get out? What does the final map show?
The direct output is an estimate of the relative DNA production rate, which the paper calls P. This P is sort of a proxy for a relative abundance or biomass of the species in each river section. But to make it more intuitive for, say, conservation planning, eDITH translates that P value into something maybe more useful. A detection probability, often called PD. So the map shows for every single reach of the river, what's the probability of detecting that species DNA if you were to sample there?
Probability of detection. That seems very practical. And you mentioned qPCR and metabar coding. Does the package handle both types of eDNA data smoothly? They can be quite different statistically.
Yes, it's designed to be flexible. If you have qPCR data, which gives you concentrations, it can use error models like the normal or log normal distribution. If you have metabar coding data, which gives you sequence read counts, it can use count-based distributions like the geometric or negative binomial. It adapts the statistics to the data type.
That's smart. And what about explaining why a species might be detected in certain areas? Can you include environmental data like water temperature or habitat type?
You absolutely can. If you have data on environmental covariates you think are important for your species, you can feed those into the model. But if you don't have that detailed environmental data, the package has a clever backup. It can automatically calculate and use something called asymmetric eigenvector maps, or AEMs.
Okay, AEMs. That sounds technical. What are they doing for the user? Are they like stand-ins for environmental factors?
In a way, yes. They are mathematical variables derived purely from the river network's structure, its connections, and flow paths. They capture spatial patterns specific to river networks, so they can act as effective proxies for underlying, maybe unmeasured, environmental gradients or processes that influence species distributions within that riverine landscape. It's a way to account for spatial patterns even without explicit environmental data layers.
Neat trick. So Did the authors test this out? Did they show it works in real rivers?
They did. They used two case studies with fish eDNA data. One was the Lower East Sydenham River in Canada. That's a fairly large system, over 1,600 square kilometers, and they had data from 30 sites. The other was the Koide River in Japan, much smaller, only about 39 square kilometers, with 11 sampling sites, so quite different scales.
And what did they find? Did eDITH manage to produce useful maps in both?
Yes, for both rivers, they ran the model independently for different fish species. And in both cases, eDITH successfully generated these spatially explicit maps showing the probability of detecting each species across the network. It also allowed them to estimate overall species richness patterns across the rivers. It demonstrated that the approach works across these contrasting systems.
That's compelling. So, stepping back, what does this mean for the bigger picture, for science, for conservation? It feels like this could really change things.
I think it has huge potential because it provides a standardized, accessible way to upscale eDNA data. It could be applied to pretty much any river system worldwide, as long as you can collect that minimal input data. This could massively improve how we assess biodiversity in freshwater ecosystems, especially in places that are currently hard to monitor or haven't been studied much. We could get much higher resolution pictures of where species are.
And it's not just fish, right? You mentioned it's broader.
Oh yeah, absolutely. The principles apply to DNA from almost anything shedding into the water. Other aquatic life like invertebrates, amphibians, plants, even microorganisms, potentially even terrestrial animals that come near the river, if that assumption holds that their DNA shedding is somehow related to their presence or density near the water, the model structure itself is quite general.
That versatility is really impressive. But powerful models usually come with caveats. What are the main limitations people need to keep in mind if they want to use eDITH?
Right, there are some important ones tied to those core assumptions. First, the model assumes the river water is well mixed vertically and horizontally at the sampling point. In really wide or complex rivers, that might not be true. So you might need to adapt your sampling, maybe take samples from multiple points across the channel width to get a representative mix. Second, and this is a big one, eDITH is specifically designed for flowing river networks. It's not built for systems that include lakes or large reservoirs.
Why not lakes?
Because the water sits there for much longer. In lakes, DNA has more time to degrade completely, or it might settle out into the sediments. That breaks the whole downstream transport and decay model that eDITH relies on. The hydrology is just too different.
Okay, no lakes or reservoirs. What else?
It also doesn't handle situations where water is moved against gravity, like with artificial pumps transferring water between basins. And it technically doesn't handle river bifurcations where the channel splits downstream, like in braided rivers. Although, for braided sections, the paper suggests you can often approximate it conceptually as a single channel for the modeling, so there's a potential workaround there.
Got it. So well-mixed flowing channels, no big lakes, no pumps. Those seem like manageable constraints for many river studies. Overall, though, this feels like a major step forward, taking eDNA from being just a presence-absence check at a point to actually mapping out likelihoods across whole river systems. That's quite a leap.
It really is. It transforms eDNA into a proper spatial mapping tool for conservation and management. Freshwater ecosystems are under so much pressure globally. They're incredibly biodiverse, but also incredibly threatened. Having a tool like this that helps us understand biodiversity patterns at this kind of resolution and potentially track changes over time, it could really help us figure out where problems are, where interventions are working, and how different parts of the river network contribute to the overall picture.
So perhaps the final thought for you, our listener, is this. If we can now generate these detailed, spatially continuous maps of biodiversity probability in rivers, how does that change how you might think about managing a watershed? Instead of just reacting to local data points, how does seeing the whole interconnected picture influence where you'd focus conservation efforts or restoration projects? It opens up a more strategic, network-level view, something definitely worth considering. Thanks for joining us for this deep dive.